is a waning happening with time. So that gives us an opportunity to identify those unique traits of the TFH cells which exist with the long-lasting protection. We do have these major questions in the lab to, of course, understand the traits of immune memory and the biology of TFH cells in those particular conditions. We are running some of the programs in these pathogens from Japanese encephalitis, in dengue, and of course with COVID-19. I will just give you a brief information that why we chose these models for addressing these questions on the TFH biology. So we talk about JEV infection and vaccinations here. This is a mosquito-borne flavivirus, which is actually transmitted by the Clolex mosquitoes, same family like dengue, yellow fever, Zika. And of course, the epidemiology says that it is actually, a majority of the problems exist in the Asian, the Pacific regions, almost 68,000 clinical cases. More important to see is the case fatality rate, which is approximately 30%. But the neurological sequel in this disease is even worse. So almost 50% of the survivors will have the permanent sequelae. So the question comes is, why we chose this model is because of the vaccine. The historical vaccine which is available for this disease started the mass vaccination campaign in this country in 2006. And that vaccine has shown good effectiveness just after the immunization, but slowly the immunity wanes down. And this is some of the data which we actually generated with the help of our clinical collaborators. So this actually allows us to address this particular question that in infection, you have a lifelong immunity. You never get the infection again, but the vaccination has some limitations. So why not use this particular model and then answer our question on the involvement of TFH cells. So what we did here is we enrolled and constituted our cohort in the northeast part of India, the difficult terrain, and that to start with the difficult terrain and early stages was something which was challenging, but we got help from all possible directions. We set up this and we were able to then look into the overall immune profile of the antibodies, particularly the T cells and the B cells, both at the polyclonal level as well as at the antigen specific level, to first understand what kind of dynamics of the immune response exist in these individuals, those who are infected or those who were vaccinated. What we actually got from these studies is two major hints. One, not everyone who is vaccinated is actually responding to the vaccine. The ratio, this particular figure is significantly high. Almost 20% of them do not make IgG in the single dose immunization. They don't show the memory B cells, but they do have some frequency of T cells and a bit of TFH cells available in them after the immunization. The second important thing, which was the original question, we found that in case of the recovery from the infection, or if we compare them with the responders to the vaccine, the individuals, those who recovered from the infection, showed a very high GC reaction. They had a high level of neutralizing antibodies, not only in the quantity, but also in the durability, which we have tested until 3 years. And these individuals constitute not the different frequency of the TFH cells specific to the virus, but they have some differences in their functional properties. So with these two initial hints, with this whole data that we were able to generate, we then looked into more details by approaching some of the transcriptomics or the metabolomics approaches and utilize them to identify some of the hits that can actually differentiate between why these individuals are responding and why the individuals who are not responding, not making T cells or antibodies, do have some of the T cells. And in case of this particular question where we show the recovery and the responder analysis, we are now finalizing our data on looking into the single cell RNA-seq and identifying what is so unique about the TFH cells in the individuals those who recovered from the infection in terms of their transcriptional signature or in terms of their functions. Something on the way that we have also shown, because we got some of the hint from these particular studies, that there is a very important coordinated response of CD4 TFH and the follicular CD8 actually is necessary for this particular vaccine to confer the protection, and that actually includes majority of the CD4 T cells to confer protection, but coordinated response of CD4 and follicular CD8 to establish the humoral responses. So it's not very straightforward still to say that what kind of cellular correlates this vaccine actually is inducing while we have protection from the infection. Of course, COVID-19 gave us an opportunity and expanded the horizon of our research, and it gave us a fantastic opportunity to address our questions on the biology of TFH while we compare the infection and vaccination, because there is much more diversity exists in COVID-19 in terms of the individuals, those who were asymptomatic, those who were mild, or those who were severe, and some of the vaccines had done really great. So we are actually utilizing this model to just looking into the traits of TFH cells in long term, and also identifying their traits in a broad protective immunity in context of the variants of concern. And this also has given us an opportunity to develop some immunological parameters and develop new tools for probing the T cells. So we were able to contribute to the field in the start of the pandemic to know about to provide the evidence about T cells that exist prior to the pandemic, and also to look into the durability of the T cell responses to this particular virus in the Indian population. We utilize, and because we were equipped with a lot of skills on the T cell biology, we also utilize that to understand how an inactivated whole viron vaccine which was produced in India is actually inducing the immune memory, and whether that immune memory is protecting or reacting against the multiple variants or not. And in between, because we were looking into a lot of these functions and the crosstalk of T and B cells, we addressed and we found a very improvised method of how a T cell function or B cell health can be investigated in an in vitro condition beyond the existing T and B co-cultures which are available, which do not allow us to go into high sensitivity with the antigen specificity. So we of course were working on this, and we demonstrated that in COVID, but also in many other pathogens from dengue, JAV, to even the bacterial infections like Mycobacterium tuberculosis. And in this duration, we also contributed in many other studies to at least set up the platform to address our questions on how these TFH cells are actually behaving in this kind of long-lasting immunity to the virus or to the vaccination. We did not end up there because with the request from the health agencies and also a lot of interest from the vaccine pharma industries, we actually made a satellite facility for helping them to evaluate their vaccines. And recently we helped them to evaluate the intranasal COVID-19 vaccine in a phase 3 trial to understand whether the booster immunization with this vaccine in a different context of the initial vaccines rolled out in the country is immunologically effective or not, whether it is capable of inducing the mucosal responses or not. So we have generated that information, and I think this vaccine has recently been awarded the emergency use license for the boosters. Quickly in the dengue, particularly we are utilizing this model because I think we all are aware that it has a very interesting a basis of antibody-mediated or antibody-mediated immunopathology in case of severity. And that is something which of course makes us attracting towards this particular model to say, what is the role of TFH cells here? Because are they good? Are they bad? Are they involved at all in that exaggerated responses of the B cells and the antibodies? Can we identify some good traits there? And while we do that, we also develop some of the tools to at least study these cells in the human populations in blood. And that's where we did some kind of epitope mapping, and now we identified a very highly immunodominant mix of epitopes from the dengue virus that can allow us to now examine and study the TFH cells in the blood samples. So what will I do now is I'll quickly go over some of the data, what we are doing in this particular study. And before I go there, I'll just give you, particularly to my young colleagues, a bit of information on dengue. So this is transmitted by a mosquito, which is basically coming from the Aedes aegypti species. The serotypes from dengue 1 to 4 add to the complexity of this particular disease. And if we talk about its prevalence, I think more important is to see these two things. Although this is an old factor of almost 4 billion people at risk, but it has continued to expand. It has continued to actually expand in its prevalence. And I think with the current climatic conditions, we can see a further upsurge may more be higher than this particular ratio. Something clinically, if we talk about this disease, majority of the infections do not develop into the severity. It's a minor fraction of less than 5%. And almost all of them, if I say other than the severity of the dengue symptoms, they remain asymptomatic. An interesting but unexplored area of dengue immunology. So what we did here is, basically we looked into some of the existing information, and if you look into those studies which talks about the immune factors in the disease severities, there are many actually. It includes immunological factors, but it includes viral factors, it definitely includes host factors, and also the how and what kind of clinical management was provided at the time of patient admission. Something which I would like to highlight here is, if we talk about the antibodies do not only cause severity in this particular infection via antibody-dependent enhancement. I think one of the mechanisms of antibodies which we started learning from dengue, but also by binding to some of the cell proteins. And that makes it more complicated because that it lead to a direct complications or the severe clinical manifestations in dengue. So that's where we started asking the question very first, do we see TFH cells in dengue, and what kind of implication they have in particularly exaggerated plasmablast responses or these high magnitude antibody responses which are not of all good quality or also includes the cross-reactive antibodies. So we did some study in a hospital-based settings where we enrolled the individuals with the different grades of disease and also with the different phases of infection, primary or the secondary infections, those who developed mild or the severe disease. The very first message, we didn't see a huge difference in terms of severity, whether you were a primary dengue or you were a secondary dengue, unlike what we have seen in the textbooks from past, I think, 2 decades. We also followed these individuals from their convalescence to also until the discharge from the hospital, and some of them are also followed up on year 1 or year 2 or year 3 basis, just to understand how at the early stage of this in memory or the TFS cells or the T cell which induce, how do they stay in a long term in these individuals, and what kind of response they do at the time of recall. So quickly, I'll just tell you that when we started, the very first question we asked, do we see CD4 T cells? Do we see their proliferation at the acute stage of disease? Yes, it was substantial. And if you compare with the healthy donors, it was quite an upsurge of proliferating T cells, CD4 T cells in dengue acute infection. And then we looked into the deep phenotyping to understand what all subsets of CD4s and the frequency of TFH exist, sharing just the glimpse of the data that if you look into this particular plot, if you see this area which is a proliferating cells, what we found is the cells those who are expressing CXCR5 do not exist much in that proliferating pool. But still, we found some of the cells those who were actually expressing PD-1. That was a bit surprising for us when we started, but it also attracted our attention to know what are these cells, those who have some kind of similar phenotype like TFH, and they are actually proliferating much more than TFH in case of acute infection. If you see here, when we looked into the fraction of these cells which are actually not CXCR5 positive, but actually proliferating, we found a substantial fraction of these cells in circulation, and not only substantial fraction, these cells do express a very high level of PD-1 as compared to TFH. So we then looked into the context of the kinetics of these cells, what kind of dynamics they have in the primary or the secondary dengue. As we looked into the TFH cells, it showed us something which is of course an expected response from an early stage of illness to the later, it goes up and then it is stabilized. And definitely from the early stage to the later stages, it has gone up in terms of its frequency in circulation. However, in a secondary dengue, it was not that fluctuating as we saw in case of the primary dengue. Interestingly, this subset, which was actually not expressing CXCR5, but expressing PD-1, do show a similar kinetics in blood like what we see in case of TFH. If I summarize that data, what we found is that in a dengue-specific memory pool, a virus-specific memory pool, the largest fraction of the cells exist are not expressing CXCR5, but they are PD-1 positive, and the TFH cells are the minor fraction. So that then allow us to answer some of the questions that what these cells are doing. Do we see these cells more in the severe conditions? Because having this much larger fraction of the cells in circulation, it definitely have some biological relevance. So we looked into the conditions like mild disease and the severe disease, and what we found is, unlike TFH, which does not show a huge difference in their frequencies from mild to severe, these cells do have a substantial increase in their presence in circulation when there is a mild disease to the severity of the condition. A very quick question then we ask, oh, they do share a phenotype with TFH. Do they also share some kind of functional traits with TFH? Are they able to provide help to B cells? So in multiple approaches, we looked into their capacity of secreting B cell help cytokines, and also to demonstrate whether they can really do a direct help to the B cells or not. So yes, they do have the capacity to secrete those cytokines, which are the B cell help cytokines like IL-21 from gamma, and we utilize this particular method which we developed on a very highly sensitive way to understand the T cell and the B cell crosstalk with as low as 90 antigen specific T cells. So that gave us a tool to address this question, whether these cells can directly give help to the B cells or not. So we sorted these populations along with some of the control populations, and what we found is these cells are enough capable, those who are actually expressing PD-1, to induce the plasma differentiation in the ex vivo conditions like the TFH cell population. So that then gave us an idea that yes, these cells not only exist in a high frequency, they do have the capacity to provide help to B cells. The question came quickly in mind, are they related to TFH? And do they have a transcriptional signature which is different than the TFH in terms of providing help to B cells? Can we identify the mechanism? How do they do this job of helping the B cells? And what kind of relations they have? So we have done the single cell RNA-seq on these subsets, particularly taking out the activated populations and also taking out some of the resting memory populations. I'm just giving you some raw data here where we actually pooled some of the donors, did the barcoding to identify specific donors, and then looked into both the transcriptional signatures, the protein expressions, and also their TCR clonotypes to understand how much related they are at these levels. What we found is a very interesting cluster of cells which further expanded our question, because we were thinking a very straightforward answer to this question, but no, that's not the case. What we found is, if you see this whole cluster, this says they all are Th1 kind of cells which do not express much of the CXCR5, but they are highly heterogeneous. So it's now making us more complexities to at least conclude this particular data set, but we have at least found some of the transcriptional signatures at the gene level where we saw that this particular fraction that you see in blue here looks like the subset of our interest, which has capability to provide help to B cells. It's transcriptionally different than the TFH, which do exist here in terms of their transcription factors or their surface markers. Importantly, which we have recently looked into, we still have to go into the details, that this subset, if you talk about the clonotypes they share with TFH is very marginal. That further help us to actually identify that they are actually clonally not so related to the TFH. They do have their unique identity, and they are much more widespread than the TFH cells in circulation. So the quick takeaways from this particular work is we have identified this particular subset which is functionally potent to provide help to B cells. They do acquire memory, and these cells have the capacity to induce plasmablast differentiation much more vigorously than the TFH cells in dengue. Important is that they are much more populated and accumulated in the severe dengue, and they do have unique clonotypes than the bona fide TFH cells in dengue. And the next question now we are addressing is to look into the mechanism of B cell help by these cells, and of course to actually demonstrate the relevance of these cells in case of dengue immunopathology. So I will flash this image, these happy faces in the